So the technique that I'm going to be talking a little bit about today is called somatic cell nuclear transfer, and it's a little bit of a simpler concept than some of the other techniques that we've been talking about recently. It involves an egg and a somatic cell, and a somatic cell is basically any other body cell that has been differentiated, so like a fibroblast or an osteoblast or anything like that. And so basically, the nucleus from the somatic cell will be taken and placed into the egg once the egg's own nucleus has been removed. And kind of the goal of this is to create an embryo from these two cells that will be a clone of the DNA from the somatic cell that was originally used. And so right now this is used to make a lot of genetic clones. It's used with making transgenic farm animals, and it also has possibilities with other therapeutic medicine as well. I'll get into some of those details later. And so kind of a quick overview of some of the procedural things before I go into the details. So first, the nucleus from the egg is going to be removed, and then the nucleus from the somatic cell will be removed and placed into that empty egg. And so once the egg kind of adopts this new nucleus, its own machinery, so like its polymerases and transcription factors, will work to replicate and express this new DNA that's present. And so once it starts dividing and the DNA is replicated and all of that, we can harvest stem cells that can be used for various medicinal treatments for diseases and whatnot. And then it says here that cells are complete genetic copies of the parent. And while that is the ideal that they're working towards, that's not always the case, as there are some complications and various problems that can arise that don't always allow for the offspring to be a complete clone of the parent. And I'll get into some of those reasons why here later. For more of the details of the procedure here, the very first thing that they do is they remove the nucleus from the egg that has been arrested in metaphase 2, and they discard that. They really don't have a need for the egg's nucleus after that point. And then they'll remove the nucleus from the somatic cell and place that into the egg. They use a pipette to break through the layers of the egg and insert that nucleus. And then in order to make sure that the two cell types are joined together properly, they shock the cell. That allows the egg to kind of recognize the new nucleus and start to treat it as its own. And then they allow that cell to incubate and to start propagating and dividing and to start growing into an embryo. And so in this video here is just kind of showing that procedure that I just talked about.
This video was prepared by my colleagues Dieter Egli and Kevin Hagen. And you see here on the left is what's called a holding pipette, which gently sucks on the egg. The egg is surrounded by a membrane called the zona. And you'll watch this pipette on the right first drill a hole into the zona, then go in and suck the nucleus out. And then another nucleus, which has been taken from, say, a somatic cell, a cell of the body, is going to be put in. So if we could start the video, please. You'll see now that this drilling pipette is going to drill a little hole into the membrane. You can maybe see a little bit of the hole right here at the next part. This pipette, you'll note, isn't really sharp like a syringe. There you can see a bit of the hole. And now the pipette's going to go in and remove the nucleus. And if you look carefully in the pipette, you'll see a line in the nucleus, which are all the chromosomes lined up. So that nucleus is going to be squirted out now because we don't need it anymore. And there we have an enucleated egg. Now the next step is to take a set of eggs like that, and I'll show you two, and then transfer into them a nucleus from another kind of cell, a fully differentiated somatic cell. So here the enucleated egg is set on the side, that it's held by this holding pipette on the left. There's drilling the little hole in the membrane. Here we go in. Here comes the nucleus from the right. The pipette goes in, and these pipettes are operated with a piezoelectric device. So you can't see it here, but it's like a little jackhammer going very quickly, like a Woody the Woodpecker, getting in there to then squirt the nucleus in. Here we'll see it again. A little hole in the zona is prepared, and now the nucleus is going to be squirted inside. So there's two examples of what's called somatic cell nuclear transfer.
Okay, so that video was basically just showing the overall procedure that I went over about removing the nucleus from the egg, removing the nucleus from the somatic cell, and then placing it into the egg there. And so there are a couple applications of this technique that I wanted to go through. There are a couple different routes that they go down, and so that's what this figure here is kind of about. In the top here, they just show natural fertilization. You start out with your egg here. The sperm comes in and fertilizes it, which allows activation, and then this embryo can start to grow and will eventually form the offspring here. Now, in reproductive cloning, they start off with the donor animal here, and typically they'll take the egg from the donor animal here, take the nucleus out, take the somatic cell from the donor as well, and then put that nucleus into the egg, allow it to start dividing and growing, and eventually put an embryo back into the donor animal here, who will carry it to term and eventually give birth to a cloned offspring. So that's kind of an overview of reproductive cloning. And then for therapeutic cloning, we have a donor patient, and then we have a different donor egg who typically comes from not the patient here. But this donor egg is enucleated, and then they take a somatic cell that does come from our donor patient here, put that nucleus into the egg, allow it to start growing and propagating, and then they can stop before the cells differentiate and use stem cells and put those back into the patient here for any treatment that they might need. Or they can allow the cells to differentiate here and then use those to put back into the patient as well. So for a little bit more into reproductive cloning here, like I said, they will often take both the somatic nucleus and the donor egg from the same animal, combine them, put them back into that donor, and allow her to carry to term an animal and give birth to what would be a complete clone of her. And so they often do this with animals that show exemplary DNA, or they have a specific trait that they want to keep within their herd, and so they'll breed for that trait so that their animals will continue to express it. And this is currently being done with a lot of pigs in China for commercial production. And then for therapeutic medicine here, this technique can be used a lot for regenerative medicine for patients that have degenerative diseases that could potentially generate stem cells, and those stem cells could then be used as a treatment to help with the degenerative disease. And they're particularly looking at this with mitochondrial DNA mutations that result in some sort of disease, because mitochondrial mutations are transmitted extranuclearly. So if we're able to take a donor egg that is unaffected and then use a somatic cell from the patient and transmit only the nucleus into this donor egg and allow that to then propagate, it will have the patient's DNA, but since only the nucleus was transferred into the egg, it won't carry any of the same mitochondrial mutations. So they're able to allow this to grow into stem cells and then put those back into the patient, as they do not carry the mitochondrial mutation. So then those cells will kind of become the dominant form in the patient, and the cells that carry the mutated mitochondrial DNA will often start to die off. And so that's kind of one way that they're using this as a treatment for this kind of disease. And then one of the articles that I looked at observed cows that were expressing this disease called leukopenia, which is a disease that lowers white blood cell count. And so they found this factor called HGCSF that increases white blood cell count. And so they wanted to express this in the cows, And so what they did was they created a fibroblast cell line that expressed that HGCSF and then used somatic cell nuclear transfer to insert that nucleus into an egg and then allow that to develop into an embryo, which they then implanted into a mother to carry to term this embryo that would eventually develop and express this HGCSF, which would eventually combat leukopenia so that they could start reproducing these cows that would not express this lowered white blood cell count. So that's kind of how they're using this technique now with the transgenic farm animals. And then so I also wanted to real quick get into some of the future of where this technique is headed, because there's still a lot about it that they need to fix or that they're looking into how to improve upon it. And so there are a lot of epigenetic markers that are causing problems for the egg when it is trying to replicate the DNA. And so they're looking into how that could potentially affect the viability of the embryos that they're creating. And then they're also trying to figure out why some of the cells just are sporadically arresting and dying after they reach the blastocyst stage. And then they're also really looking into improving culture conditions for gene expression by the egg, just seeing what leads to optimal replication and transcription of the new DNA once it's implanted into that egg. And so sometimes the egg will just stop reading the somatic DNA, and again, that's kind of why they're looking at what is the optimal culture for this cell. What do we need to put into it to allow it to grow quickly and efficiently? And then, as I was saying with the epigenetic markers, a lot of the somatic cells that they use express certain epigenetic markers that need to be erased in order for the oocyte to properly read the new DNA and produce kind of a clean copy of a stem cell. And so they're looking into how that may affect embryos. So that's just kind of a quick overview of somatic cell nuclear transfer.